So the purpose of this experiment is to investigate the antimicrobial activity of two different types of spray cleaners. So I have one that's Meyers Everyday Multi-Surface Cleaner Basil Scent. And then I have LYSOL All-Purpose Cleaner with Mango and Hibiscus Scent. So we're looking at the effects of these two spray cleaners that we might use like on a countertop or some other hard surface as antimicrobial agents against two different types of bacteria, one of which is a spore former, so this is Bacillus, and the other of which is going to be E. coli, which is a non-spore former. Now, big mea culpa, I did not go and buy these until yesterday, and given the coronavirus outbreak, as you might imagine, trying to find any sort of spray cleaner was next to impossible. So I did not get what I had hoped to get, which was Simple Green. That tends to be the most common, what we'll call natural cleaner, and Lysol with bleach, which is kind of a harsher version. It has bleach in it. The only things I could find, and I went to a couple different stores actually, was something akin to Simple Green. This is Meyers Clean Day versus Lysol, which looks like it has a quaternary compound in it, but it doesn't have bleach. So this is what I have. Wasn't quite what I had envisioned, but given the circumstances, I didn't get to be choosy. And then of course for control, we'll mix E. coli and Bacillus with some water. So the experimental procedure is going to be to take some test tubes where I mix either Bacillus, the spore former, or E. coli, the non-spore former, with one of the cleaners or water. And then what we do is we take a look at we'll do serial dilutions and try to figure out what the concentrations are. Now, the first thing to note is that when designing this experiment is I need to know about how much cleaner would I put into a test tube versus some sort of broth. Now the broth is pretty concentrated, and so if I were to do something like a 1-to-1 ratio where I did 1 ml of broth and 1 ml of cleaner, that may or may not approximate what you would encounter in real life. Since these are used mostly on surfaces, and surfaces have usually only a small amount of water on them, if any, and unlikely to have huge concentrations of E. coli or Bacillus. The exception is that if you're in the bathroom and somebody's had a major GI infection, you might actually be cleaning up some liquid feces, and then this wouldn't hold true. You might actually want to break out the bleach for that. But since we're looking more like at surfaces, just kind of standard cleaning, I'm going to do 1/2 ml of each of these cultures with 1.5 ml of cleaner or water, because I feel like that's probably a more realistic ratio that we'd experience on a countertop. So to get started, I'm actually just going to do my mixes. So I'm going to go ahead and get my test tubes, label them. I'm going to have 6 test tubes in total. I'm going to have each a Bacillus and an E. coli, and each of those are going to have 3 test tubes. Bacillus is going to have one that has water, a second test tube with Bacillus and Lysol, and a third test tube with Bacillus and Meyers Cleaner. And then the other ones are going to be E. coli, and I'm going to have E. coli and water, E. coli and Lysol, and E. coli and Meyers Cleaner. Now, at this point, I'm actually going to stop the, in a moment I'm going to stop the recording, and then I'm going to do a kind of a rapid speed play of what I'm doing. Now, the reason why I'm going to stop is because I can't do all 6 tubes right now. If I were to inoculate Bacillus and Lysol, then it could be 20 minutes before I actually do the serial dilutions of that tube. And the reality is nobody leaves, well, I shouldn't say nobody. Few people are leaving Lysol on their countertops for 20 minutes. So again, to approximate more realistic times, I'm going to try to make a combination and let it sit for exactly 1 minute before starting my serial dilutions. So what you're going to see is me doing a set of tubes, one tube, and then immediately going into the serial dilutions, putting it away, a second tube, doing the serial dilutions, putting it away, where each time, as soon as I add the Bacillus and the Lysol together, or the Bacillus and the Meyers, or E. coli and Lysol, or E. coli and Meyers, they are not allowed to interact for more than a minute before I do a serial dilution. And by doing a serial dilution from a test tube where it's all concentrated into the 99 ml water bottle, that effectively stops the reaction of whatever might be killing bacteria, because I dilute out the cleaner so much that there's just no way that it's really going to work at that type of dilution. Okay, so let me go ahead and get started. Okay, so what I'm doing now is I'm taking 1.5 mils of each cleaner and water and putting them in my test tubes. Okay, so now that I have my test tubes, they'll have 1.5 mils. Again, I'm going to work through just one test tube at a time, adding either E. coli or Bacillus to it, and then waiting a minute and then doing a series of serial dilutions. So I'm only going to do a couple of these for you to watch, otherwise you're going to watch me do this 6 times and you're going to get really bored with that. All right, I'm going to try to move this a little bit and get a better view. Okay. So first I'm going to set up my bottle A, B, and C. And I'm going to go ahead and start with E. coli and water, which is this tube right here. Okay. So we're going to do a series of serial dilutions with bottles A, B, and C, just like we did in Lab 8. So we're following Lab 8 protocol almost exactly for at least the water one. The other ones you'll see that I'll do some deviations to them. Okay, so I'm getting my pipette. I'm going to take 1/2 ml of the E. coli solution, which is pretty well mixed already, and I'm going to add it into the tube that says E. coli and water. And then I'm going to start my clock for exactly 1 minute. Okay, so while I'm waiting for that 1 minute to happen, I'm going to get my bottle A set up here. So I have my bottle A, and I'm going to have B and C too. And then exactly 1 minute from when I started, I'm going to take 1 ml from this and put it into bottle A. And once I do that, I have a little more time. Okay, I still have 20 more seconds. I'm going to go ahead and get ready. 10 more seconds. And here we go. Oops. 1 ml into bottle A. Okay. So we're now done with this tube. I'm going to move it off to the side. I'm going to move the rest of my tubes off to the side, And now I'm going to go ahead and get ready to do my serial dilutions for E. coli and water. Okay, I have my setup now. So I basically have my 3 bottles, bottles A, B, and C, like you would in a typical lab A procedure. I have my 4 Petri dishes in front of me. I have, and I've written on them like E. coli plus water, 10 to the minus 4. I have a 10 to the minus 5, 10 to the minus 6, and a 10 to the minus 7. And I'm setting this up just like you would for lab 8. Like I said, once you put your sample into bottle A, you have not an unlimited amount of time, but you are not constricted to the same time frame as you were when you're waiting for that initial test tube to be diluted out into bottle A. That has to be timed exactly at 1 minute, where the rest of this, you've got up to half an hour probably to do your serial dilutions here. All right, so I'm going to go ahead and do the set. Okay, so now what I'm going to do is go get the auger to make pour plates. Okay, but I also want to talk about what I'm going to do for the other 5 tubes. So Bacillus, I'm going to do the exact same thing that I just did for E. coli. I'm going to follow lab 8 procedures, and I'm going to do serial dilutions to the 10 to the minus 4, 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7 plates. Now, I'm not exactly going to follow those dilutions for my other samples, the ones that have cleaner in them, like LYSOL or Mrs. Meyers. Yeah, it's Mrs. Meyers. I had to check. And the reason is because I actually think that especially LYSOL should kill some of the bacteria. So if it kills too many of the bacteria, and I do a 10 to the minus 4, 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7 dilutions, it's very possible that I won't have any colonies at all. So what I need to do is actually kind of estimate where I think the countable plate is going to be and scale back my dilutions accordingly. So and this is just me kind of guessing where I think it's going to land based on previous experiments. I'm probably going to run the E. coli using a 10 to the minus 2, a 10 to the minus 3, and a 10 to the minus 4. And for Bacillus, I'm going to do a 10 to the minus 3, a 10 to the minus 4, and a 10 to the minus 5. So I am about to do a serial dilution of Bacillus and LYSOL. And unlike the previous dilutions that I did with water, where I followed Lab 8 protocols exactly, I'm making some modifications here. So first of all, you might notice that I only have 2 bottles out, A and B. I don't have a bottle C. That is because I expect that the Lysol will actually kill a lot of the cells. And so if I go out too far in my serial dilutions, I'll end up with empty plates with 0 or 1 or 2 colonies on them. And of course, I want to hit the plate with between 30 and 300 colonies. So I'm moving my serial dilutions back to the more concentrated realm. I'm going to do a 10 to the minus 2 dilution out of bottle A. I'm going to do a 10 to the minus 3 dilution out of bottle A and a 10 to the minus 4 dilution out of bottle B. So here I go. So like last time, I'm first going to draw out half a milliliter of the Bacillus and then mix it with my Lysol for exactly 1 minute. The Bacillus is a little chunky. You might recall that this species forms a really solid pellicle at the top. So I'm pipetting it up and down more just to make sure that the chunks are more evenly dispersed so we can get accurate counts. And here I go. Okay, so that stops the reaction of whatever in Lysol might kill the cells. It's been diluted out too much in this 99 ml bottle. So now I've bought myself a little bit of time. So in lab 8, you guys did not do a 10 to the minus 2 or a 10 to the minus 3 dilution. But if you understand how serial dilutions work, I can pull 1 milliliter out of this bottle A and put it in this plate, and this is a 10 to the minus 2 dilution. Recall that 10 to the minus 2 is the same thing as 1 in 100, or 1 over 100, and since this is a 99 ml bottle, and I took 1 milliliter of this solution and put it in this 99 ml bottle, that was my 1 in 100 dilution. So now I'm just sampling that, putting it on a plate, and I've basically taken a milliliter of that 1 in 100 dilution. Likewise, to do a 10 to the minus 3 dilution, I'm also going to use bottle A, but instead of pulling out a full milliliter, I'll only pull out 0.1 milliliters. Okay, so now I have the drops on my plates. I'm going to do the same thing over here for E. coli. I'm going to add agar to everything to make pour plates, and I'm done with the experiment. Hey, so it's the next day, and we are looking at the results of our pour plates. And so I have unstacked the pour plates, and I've started to take a look at them. And so what I'm going to do now is I'm going to actually just take photos of the pour plates, and I will go ahead and embed them into this video. You will now be presented with the results of the pour plates. In each case, a set of plates will be shown first as an image, and then it will transition to each individual plate being pulled to the foreground, where you will be responsible for following the Sharpie marks to determine the number of countable colonies on each plate. In cases where there are too many to count via this technique, I will usually fade away and then bring the picture back with the number of colonies that are marked in each half or quadrant. If there are too many to count, you will see that I will write TNTC, too numerous to count. For the E. coli and Mrs. Meyers set of plates, all of the plates ended up being too numerous to count. That said, we can still attempt to count the highest dilution, which is the 10 to the minus 5. It's not as accurate as the count would be if the colonies were numbered between 30 and 300. But since I do not have a 10 to the minus 6 dilution to count from, I'm going to have to estimate the number of colonies on the 10 to the minus 5 plate and use that for my calculations.